Okay, so amino acid biosynthesis. Plants and microorganisms, they're able to make all 20 amino acids. Um, bio amino acid biosynthesis. In amino acid biosynthesis, glutamate, okay, glutamate is the primary source of nitrogen via transamination. Okay, and how this happens is you have your glutamate plus an alpha keto acid. Wow. And an aminotransferase enzyme, which catalyzes the reaction for a new amino acid to be synthesized. Okay, so I'm not going to get too much into the mechanism of this. The only thing that I really want to mention is that amino acids are grouped into different families according to the metabolic intermediates that serve as their progenitors. So the most basic of these are the alpha ketoglutarate family amino acids, and as we recall, alpha ketoglutarate is part of the citric acid cycle. So these in general are glutamate, glutamine, proline, arginine, and in some cases lysine. Lysine is only in yeast and some protists like euglena. So this is not in humans. Okay, another one that I want to mention are the amino acids from the pyruvate family, and these include alanine, valine, and leucine. Okay, so like back to the aminotransferases. Aminotransferases are named according to their amino acid substrates. So, for example, if you were synthesizing aspartate, you would have an aminotransferase that is named glutamate aspartate aminotransferase. Okay, so this gives you an idea of what is being synthesized. So the way that aminotransferase reaction works is that it provides a general means of exchange of nitrogen between amino acids and alpha keto acids. The vital reaction is catalyzed by pyridoxal phosphate. That's pyridoxal phosphate, PLP, which is the active form of vitamin B6. Okay, so as I mentioned before, the amino acids that are derived from alpha ketoglutarate are glutamate, glutamine, proline, arginine, and in some cases, lysine. So proline, I'm going to focus on proline. It's derived from glutamate, and it undergoes a series of 4 reactions, and these include activation, reduction, formation of a shift base, and then an NADPH, which is the most important, NADPH dependent reduction, which ultimately gives proline. So results in proline. Arginine biosynthesis is also important to know. It involves enzymatic steps that are part of the urea cycle. And the urea cycle is a metabolic pathway that allows certain animals to excrete any excess nitrogen arising from overconsumption of protein. So, net synthesis of arginine depends on the formation of ornithine. Okay, so in order for arginine to form, we need ornithine. Okay, and ornithine is derived from glutamate, and the reaction pathway resembles the proline biosynthetic pathway, and the way is it's acetyl-CoA dependent reaction, and an ATP dependent phosphorylation of N-acetylglutamate gives N-acetylglutamate 5-phosphate, and then this primes the substrate for a reduced pyridine nucleotide dependent reduction to a semialdehyde, and then the semialdehyde is aminated by a glutamate dependent aminotransferase, which gives acetyl-ornithine and acetyl-ornithine, and then this is deacylated to ornithine. So in mammals, ornithine is made from glutamate, and it occurs via a pathway that does not involve an N-acetyl block. Ornithine has 3 metabolic roles. The first one is that it serves as a precursor to arginine. The second one is it functions as an intermediate in the urea cycle. And then the third is to act as an intermediate in arginine degradation. So it is important to note that ornithine in the urea cycle gets converted into citrulline by an enzyme known as CPS1. It's an ATP-dependent enzyme. And the CPSI enzyme, it helps to eliminate excess NH3. And it does so by breaking down arginine. So the nitrogen excretion mechanism, it occurs in the livers of terrestrial vertebrates. And another important thing to know is that the urea cycle is linked to the TCA cycle by fumarate.